Zo te gaan staan. Ik heet eerst, om te starten, iedereen van harte welkom bij deze viering. En hierbij open ik officieel deze academische zitting, waarbij Jette de Vos haar academisch proefschrift getiteld Body, Brain and Emotions, the interplay between peripheral and central neural processes in the subjective experience of emotions, in het openbaar gaat verdedigen. Mag ik u vragen een samenvatting te geven van die proefschrift in de komende 15 minuten. En ik geef u graag het woord.
Ja, zeker. Beste prorector, leden van de promotiecommissie, collega's, familie en vrienden. Dank jullie wel dat jullie er allemaal zijn vandaag. In de komende 15 minuten zal ik jullie meer vertellen over het onderzoek wat ik de afgelopen jaren heb gedaan en wat heeft geleid tot het proefschrift getiteld Body, Brain and Emotions, the interplay between peripheral and central neural processes in the subjective experience of emotions, of in het Nederlands lichaam, brein en emoties, de wisselwerking tussen perifere en centrale neurale processen in de subjectieve ervaring van emoties. Laten we beginnen bij de vraag, wat zijn emoties? Dat is een vraag die al jaren wordt gesteld door onderzoekers uit verschillende disciplines, maar waar eigenlijk nog altijd geen eenduidig antwoord op is. Waar de meesten het wel over eens zijn, is dat emoties gepaard gaan met een subjectieve ervaring, dus het gevoel van ik ben bang of ik ben blij, met lichamelijke reacties. Dus als je gespannen bent, zullen jullie herkennen dat bijvoorbeeld je hartslag hoger is of dat je klamme handen hebt. En als laatste dat er veranderingen zijn in het brein. Bij emoties zijn bepaalde gebieden meer actief en andere gebieden in het brein zullen tijdens emoties minder actief zijn. Maar er is nog veel onbekend over hoe deze verschillende componenten van emoties precies samenhangen en hoe ze elkaar en de subjectieve ervaring van emoties beïnvloeden. Hoe beter we al deze associaties begrijpen, hoe beter we kunnen ingrijpen als er ergens iets misgaat in deze emotionele processen, wat het geval is in veel mentale gezondheidsproblematiek. Het doel van mijn thesis was dan ook om door middel van verschillende methodes, manipulaties en metingen ons inzicht in de relaties tussen componenten van emoties te vergroten. Veel onderzoeken die wij doen vinden plaats in een gecontroleerde onderzoekssetting. Om dan een bepaalde emotie te kunnen bestuderen, moeten we er eerst voor zorgen dat deze emotie aanwezig is. Om dit te bereiken bestaan er verschillende methodes om in een onderzoekssetting emoties te induceren. Daarnaast bestaan er verschillende manieren om deze emotieinductie te manipuleren en zo te bestuderen hoe dat de uiteindelijke emotie beïnvloedt. En om dan daadwerkelijk te meten wat het effect van deze methodes en manipulaties is, kunnen wij verschillende metingen doen. En voorbeelden van metingen die wij in het huidige proefschrift gebruikt hebben, zijn vragenlijsten, metingen van lichamelijke reacties, zoals bijvoorbeeld de hartslag, en metingen van de hersenactiviteit. In ons geval hebben we gebruik gemaakt van de MRI-scanner. Dan ga ik nu iets meer in op de verschillende studies die we hebben gedaan in deze thesis. En bij de eerste 2 studies hebben we gekeken hoe manipulaties van lichamelijke processen de subjectieve ervaring van emoties beïnvloeden. Een voorbeeld van een intense emotie die gepaard gaat met duidelijke subjectieve en lichamelijke componenten is paniek. Tijdens paniek is er een plotselinge en intense angst die gepaard gaat met bijvoorbeeld hartkloppingen, zweten en kortademigheid. Het inhaleren van verhoogde percentages CO2 is een aangetoonde methode om een paniekreactie te induceren. Ter vergelijking, in de lucht die wij inademen zit ongeveer 0,04% CO2 en in de onderzoekssetting gebruiken wij lucht met 35% CO2. In de afgelopen jaren is er veel onderzoek gedaan naar deze methode van paniekinductie en is er onder andere onderzocht wat de effecten zijn van farmacologische manipulaties op deze paniekinductie. Deze onderzoeken breiden zowel onze kennis uit over de mechanismes van paniek als ook over de mogelijke effectieve medicamenten bij paniek. In hoofdstuk 2 van het proefschrift hebben wij alle onderzoeken die dit hebben onderzocht verzameld, om zo de resultaten uit deze onderzoeken te kunnen combineren en tot een overkoepelende conclusie te kunnen komen. Deze methode heet de meta-analyse en hiermee kunnen er betrouwbare conclusies worden getrokken dan wanneer we naar de resultaten uit één losse studie zouden kijken. We zagen dat de meeste studies hadden gekeken of een farmacologische manipulatie de reactie op de CO2-inhalatie konden verlagen. En dat bleek inderdaad zo te zijn. Daarnaast heeft een deel van de studies gekeken of een farmacologische manipulatie ook de reactie op de CO2-inhalatie zou kunnen versterken. Dat bleek echter niet zo te zijn. Een mogelijke verklaring voor de afwezigheid van dit effect is dat de door CO2 geïnduceerde paniekreactie al zo intens is dat het moeilijk is hier ook nog een versterking in aan te brengen. In hoofdstuk 3 hebben wij vervolgens zelf een onderzoek uitgevoerd met de farmacologische manipulatie en de CO2-paniekinductie. En specifiek hebben wij farmacologische manipulatie gebruikt die lichamelijke componenten van de reactie op de CO2-inhalatie zou moeten verminderen. En hiermee wilden we onderzoeken wat het effect hiervan was op de subjectieve ervaring van de paniekinductie. Zoals verwacht zagen wij een verlaging in de hartslag en de bloeddruk in proefpersonen na het innemen van deze farmacologische manipulatie. En deze verlaging zagen wij niet in proefpersonen na het innemen van een placebo-pil. Maar als we vervolgens keken naar de subjectieve ervaring van de emotie, dus als wij keken naar antwoorden die proefpersonen gaven op vragen als hoe angstig voelde je tijdens het meest intense moment van inhalatie, of hoe oncomfortabel voelde je tijdens het meest intense moment van inhalatie, dan zagen wij geen verschillen tussen de proefpersonen na het innemen van de farmacologische manipulatie of na de placebo. In deel 2 van het proefschrift hebben we vervolgens gekeken hoe het brein zich gedraagt bij emoties. En specifiek hebben we gekeken hoe het brein zich gedraagt bij aangeleerde emotionele associaties als gevolg van een proces genaamd klassieke conditionering. Bij klassieke conditionering is er een neutrale stimulus, zoals bijvoorbeeld een parkje in de buurt, die gekoppeld wordt aan een stimulus met een bepaalde emotionele waarde, bijvoorbeeld een bijtende hond. Hierdoor zal deze neutrale stimulus ook een bepaalde emotionele waarde krijgen en deze stimulus zal hierdoor ook in de afwezigheid van de bijtende hond een gevoel van angst en ongemak kunnen opwekken. En dit proces van klassieke conditionering wordt geacht als een van de onderliggende mechanismes van klinische gevoelens van angst. En dit proces kunnen wij nabootsen in een onderzoekssetting, bijvoorbeeld door het vervangen van het parkje door een geometrisch figuur en de bijtende hond door een elektrische schok. Hierdoor zal uiteindelijk ook het geometrische figuur een gevoel van angst en ongemak kunnen opwekken. In de behandeling van klinische angst wordt vaak gebruik gemaakt van exposure-therapie, waarbij de persoon herhaaldelijk wordt blootgesteld aan de stimulus die nu als angstig wordt ervaren, zonder dat de gevreesde uitkomst aanwezig is. Bij genoeg herhaling zal de angst die deze stimulus oproept afnemen. En ook dit proces kunnen wij in de onderzoekssetting nabootsen en dit wordt ook wel extinctieleren genoemd. In hoofdstuk 4 van de thesis hebben wij onderzocht hoe de communicatie tussen en binnen netwerken in het brein verandert tijdens het aanleren van deze associatie, om zo meer inzicht te krijgen in de onderliggende mechanismes bij het ontstaan van angstassociaties. Een netwerk is een groep breingebieden die vergelijkbare activatiepatronen laten zien. Met deze analyse vonden wij dat de connectiviteit in het default mode netwerk, dat is een netwerk wat betrokken is bij bijvoorbeeld het genereren van interne gedachten, verandert tijdens het aanleren van deze emotionele associaties. En specifiek zagen wij dat bij stimuli waarbij de proefpersonen leerden dat ze schok voorspelden, dit netwerk na een tijdje op non-actief werd gezet. Terwijl het juist op actief werd gezet bij stimuli die geen schok voorspelden en dus veilig waren. Opvallend was dat in een groep proefpersonen met hogere mate van affectieve klachten dit netwerk zowel bij de veilige als bij de onveilige stimuli op non-actief werd gezet. In hoofdstuk 5 hebben wij vervolgens gekeken of kenmerken van hoe iemand reageert op extinctieleren, zowel gemeten met vragen als metingen in het brein, kunnen voorspellen hoe goed iemand reageert op exposure-therapie. Ondanks dat exposure-therapie een van de meest effectieve behandelingen is bij klinische angst, zijn er nog altijd veel mensen die hier niet genoeg baat bij hebben. Dus als wij beter kunnen voorspellen welke mensen hier wel en welke mensen hier minder baat bij hebben, kunnen er betere keuzes worden gemaakt in het opstellen van een behandelplan van zo iemand. Eerder onderzoek liet zien dat kenmerken van extinctieleren de korte termijn effecten van exposure-therapie konden voorspellen. En in hoofdstuk 5 hebben wij vervolgens gekeken of dit ook gold voor de lange termijn. Helaas zagen wij dat kenmerk van extinctieleren deze lange termijn effecten van exposietherapie niet konden voorspellen. Dus meer onderzoek is nodig om te kijken of er andere factoren zijn die deze lange termijn therapie-effecten wel kunnen voorspellen. In de laatste studie hebben we gekeken naar een gebied in het brein dat belangrijk is bij het representeren van lichamelijke processen en veranderingen hierin. Deze centrale representatie van lichamelijke processen wordt ook wel interoceptie genoemd. En de insula wordt gezien als het belangrijkste hersengebied als het gaat om interoceptie. In dit laatste deel van het proefschrift hebben we gekeken of mensen de hersenactiviteit in de insula kunnen controleren door middel van neurofeedback-training. Neurofeedback-training kun je een beetje vergelijken met het aflezen van je hartslag op een smartwatch. Veel mensen hebben tegenwoordig zo'n smartwatch en hierop kun je zien of zul je zien dat je hartslag rustig is of laag is als je rustig wandelt en omhoog schiet als je bijvoorbeeld een sprintje trekt. Na het trekken van zo'n sprintje kun je ook weer proberen je hartslag omlaag te brengen. Bijvoorbeeld door een paar keer diep in en uit te ademen. En door dan vervolgens op je smartwatch af te lezen hoe hoog je hartslag op dat moment is, kun je zien of deze techniek van in- en uitademen effect heeft op de hartslag. Hetzelfde principe wordt eigenlijk toegepast bij neurofeedback. Bij neurofeedback heeft de proefpersoon bijvoorbeeld de opdracht om de hersenactiviteit van een bepaald hersengebied omhoog of omlaag te brengen. En terwijl de persoon dat doet, kunnen wij meten hoe actief dat gebied daadwerkelijk is en deze informatie terug aan de proefpersoon geven. Met deze informatie kan de proefpersoon dan vervolgens aan de slag of op zoek gaan naar methodes of manieren om deze hersenactiviteit te sturen. In hoofdstuk 6 hebben wij een paradigma getest waarin proefpersonen neurofeedback training voor de insula kregen en we hebben in eerste instantie een paar aspecten van het paradigma getest op haalbaarheid. Zo hebben wij onder andere getest of mensen de insula op commando konden activeren. Net als wanneer ik jullie nu zou vragen om je hartslag omhoog te brengen. Dit was de proefpersoon inderdaad gelukt. Daarnaast hebben we getest of proefpersonen de insula ook tot een specifiek niveau konden activeren. Dus net als ik jullie nu zou vragen je hartslag omhoog te brengen naar bijvoorbeeld precies 90 of precies 120 slagen per minuut. Dit vraagt namelijk om meer controle over deze hersenactiviteit. Dit was de proefpersonen echter niet gelukt. Samengenomen legt deze studie een basis voor verder onderzoek naar regulatie van de insula en de invloed hiervan op de communicatie tussen het lichaam en het brein. Samengenomen dragen de onderzoeken uit het proefschrift bij aan ons inzicht in hoe de verschillende componenten van emoties zich tot elkaar verhouden. Door het manipuleren van en in te zoomen op een deel van deze componenten en te bestuderen hoe dat andere componenten van emoties beïnvloedt, hebben we de kennis op dit gebied uitgebreid en paden uitgestippeld voor vervolgonderzoek. Zo vonden we dat het inhiberen van de lichamelijke componenten van paniek geen invloed had op de subjectieve ervaring van paniek. Daarnaast vonden we dat er veranderingen plaatsvinden in de communicatie in het default mode netwerk bij het aanleren van angstassociaties en dat individuele verschillen in het uitdoven van deze associaties in het lab niet goed konden voorspellen hoe iemand op de lange termijn reageert op therapie gericht op het uitdoven van deze angst. En als laatste vonden we dat mensen zelf hersenactiviteit die betrokken is bij interoceptieve processen kunnen sturen. Toekomstig onderzoek kan deze methode gebruiken om de associaties tussen het lichaam, het brein en emoties verder te onderzoeken. Hoe beter we al deze associaties begrijpen, hoe beter we kunnen ingrijpen als er iets misgaat in deze emotionele processen, wat het geval is in veel mentale gezondheidsproblematiek. Maar uitbreidingen van de huidige onderzoeken met meer metingen, meer manipulaties en andere emoties dan angst en paniek zijn nodig om deze associaties volledig te kunnen begrijpen en te beïnvloeden. Dan wil ik jullie bedanken voor jullie aandacht en ik geef het woord graag terug aan de prorector.
Thank you very much. And as you can understand now already, it is on request from the promotion dean that the rest of the thesis defense will be in English. And before we start, I'm going to introduce 5 persons. First of all, myself, I am the pro-rector. I'm Professor Steinbusch, Emeritus Professor of Neuroscience, and I'm assisted today by Professor Schumann, who is my secretary today. And of course, more importantly, we have a promotion team who's sitting opposite of me, and I start with the first promoter, Professor Linden. He's professor in translational neuroscience, but also the scientific director of the Institute Mens. Adjacent to him is Professor Schuurs. Schuurs, he is professor in mechanisms of affective disorders of our university, and also the third member of our team is Dr. Leibold, and she's also affiliated with the Department of Translational Affective Neuroscience, and that's basically the team who has guided you in the last 5 years till this afternoon that you're standing here. Welcome. Also for information here into the audience, we have 4 members who are here on site, and we have 2 members today online who will appear on the screen. You don't have to look there, they will appear on the big screen once they ask questions. And the first one who already starts to appear on the big screen is Professor Rutten. Professor Rutten was the chair of the assessment committee, and he is professor in neuroscience of mental illness, and I would like to give him the opportunity to start the discussion with you.
Thank you, Mr. Prorector. Dear candidate, my great compliments to your work. It was a complete pleasure to read your thesis. I read it with great interest. It was filled with multiple approaches from a very difficult field of research. It's experimental human neuroscience research, and it's a great tradition, I think, in your promotional team to execute such studies. And you have done a wonderful, you are a wonderful example of students doing this really, really well and really making use of multiple approaches and research methods, including meta-analysis, pharmacology, MRI imaging, and complicated statistics. So big compliments to you and your promotional team. I would like to pose a few questions because I've read it with great interest, but I also was puzzled by a few things. And I want to start off with a question relating to Chapter 2. Chapter 2 is the meta-analysis chapter. And on page 42 of this chapter, you wrote that you performed meta-analysis and you showed an overview of the meta-regression model with medication target, the biological target, as moderator. And I find that conceptually very interesting because you would like to explore a bit what type of biological manipulations can we perform and how can we help people with panic attacks. So on page 42 is in figure 2, you've showed the overview of the different biological targets, and you've analyzed them with your meta-analysis of, what was it, I think 35 studies and approximately almost 1,000 research subjects. But I think for experimental studies, that's quite large from these different studies. And you've been trying to interpret the findings of your meta-analysis, and you focused on your analysis on the different biological targets. But I was looking at this table, that's my question for clarification, that's my first thing. So there are medication targets or biological compartments like dopamine, glutamate, histamine, where there's only one small study performed on this biological target, and you incorporated them into the analysis. Now I was wondering, I would always expect that you don't find the difference because you have one small study, and I was wondering what are your thoughts about this, and was it really worthwhile to really differentiate these different biological targets because you ended up with a null result, right? So a question for clarification.
Yeah, highly esteemed opponent. Thank you for your nice words and your compliments to us and the team. And thank you for your interesting question. So if I understood correctly, your question is mainly about why including all those studies, even if there's just 1 or 2 in some categories. So I will try to clarify that. So the graph on page 42, or table, it is actually a significant result. So looking at the meta-analysis, so let me start. We did 2 things in the meta-analysis. One analysis was taking all those studies together that aim to inhibit the panic response and see if altogether with this meta-analysis we see a significant reduction of the panic response, and that we do find. And then indeed, we also compared the pharmacological targets. And as you can see in this graph, some pharmacological targets just have one study grouped there. So what we did there, we did indeed run a meta regression to disentangle results of one of the categories, but we only did this for the GABA and serotonin category, and then the rest. Indeed, we didn't think it would be worthwhile to test if, for example, dopamine, the dopamine category had a significant effect, because that's just one study. So all those small categories we grouped as one. So that was just the other category, because with this we didn't aim to say something about those categories, but more to see, at least for serotonin and the GABA category, if those two categories had a distinct effect compared to the other or the serotonin or GABA category. I hope that's
excellent. Excellent answer. I was entirely clarifies my question. It's a very good answer to this. So next point would be looking at all your chapters. I mean, I really find this really bold that you did this experimental studies, and then you have quite small sample sizes relatively in your studies, which are big for experimental human studies, but in essence they are quite small, like chapter 3, 54, and chapter 4, a little bit more than 100, and chapter 5, 43, at follow-up 33. So sample size is an issue, of course, in trying to interpret the results. And I was thinking, when you're now in the phase of looking back at your studies and thinking, okay, what is really robust in my findings, and how, I hope that you also have been thinking about this, how would I have done it if I would be back at the starting point? And I was thinking, is it, if you would redesign your own studies, is it based on sample size that you think maybe we should have bigger sample sizes? Is it heterogeneity of the research subjects in the groups? Is it the precision of your phenotypes or the type of phenotypes that you've chosen? Because I think overall you had mostly no findings, which was, I think it's very nice, but normally you have particularly in experimental neuroscience, you have a hypothesis which you want to falsify, and then you hope to find a significant or statistically significant difference. And you had some findings of interest, but most of it were maybe disappointing for you, not for me as a scientist, and also maybe not for you. So I was, the open question would be, looking back at your entire thesis, and particularly those experimental studies, chapter 3, 4, and 5, what would you have altered retrospectively looking at them?
Yeah, thank you for the very important question. I think it's a very big question to answer, but let me start and see where we get.
You can just have a top two or so, priority 1 and 2.
So I think one of the first questions was, do I think they are robust or reliable, the results, considering the sample sizes? So if we take, for example, chapter 4, with the connectivity, I think, well, that's also the one with the largest sample size, so maybe that's a bit biased, but I do think that one is quite robust. Also, if we compare it with results from other studies, for example, finding that this default mode network work is present or is changing in those kind of studies. So that may be part one. If I would have liked a bigger sample size or change something differently in the future, I think, for example, for study chapter 5, this is indeed pretty small sample size, especially for the analysis that we do. Which is the way it is for now. But indeed, it would be very interesting also because, especially this study would have has, I think, the most clear clinical implications. The findings that we have there, if we try to predict those exposure therapy outcomes. So those results, I would be very interesting to replicate or test again in a bigger sample. And maybe also to see if there's enough variation also after 1 year, and really to see if the extinction characteristics can or cannot predict those long-term exposure therapy outcomes. Although again, if we do look at why do we find or do not find this predictive value of extinction characteristics for the long-term exposure therapy outcome, when we thought about it, I do think it makes sense because in a year a lot can happen. So I don't think it's that strange that just one measurement of task indices doesn't predict the whole year of events or how things turn out. But yeah, I would be very interested to see in a bigger sample what we find there.
So hearing this, I think that's a wonderful start of the opposition and particularly the defense. With that, I think the next speakers will take over asking you critical questions about your thesis. And I would like to give the word back to the prorector.
And then we have to proceed swiftly to our next opponent, is Professor Hermans. He was also a member of the assessment committee and he's professor in cognitive affective neuroscience at the Radboud University in Nijmegen. And thank you for joining us today.
Thank you, Prorector. Dear candidate, it's a pleasure for me to be here. It's also kind of special. I say it's always a pleasure to read a thesis, but in this case it's more special because you're of course a former student at the Donners, and we've come to know each other quite well, and then you left a couple of years back. Actually seems like a very short while ago, and now suddenly I'm sitting here and you produce this beautiful book, while we are actually still in the process of publishing your master's thesis. So maybe it says more about us than about you. But it's really a magnificent work you produced here with really a variety of different pharmacological manipulations, experimental paradigms, measurement techniques, fMRI, neurofeedback, everything. So really many different things and beautiful, important conclusions you draw from this. So my compliments for this and also to your supervisors. But I'll stop short of actually congratulating you because you still have to do your defense, as you know. So I have a couple of questions, and I will start with chapter 3. And in chapter 3, I think you do a very elegant study, particularly regarding the pharmacological manipulation, where you have this comparison between a peripherally acting, centrally acting beta blocker, to try to understand how this affects the experience of emotion during a panic induction. Now, maybe the results are a bit disappointing because you don't really find an effect on subjective affect, subjective emotion, right? Now, I know you to be quite modest, but you do draw a pretty strong conclusion here, where you said information from peripheral and central noradrenergic system or adrenergic system does not play a major role in the construction of subjective emotion. So I found that quite a strong conclusion because you have a null finding. So what should we make of this? Do you think your null finding is sufficiently strong to count as evidence against the idea of noradrenaline being involved in subjective emotion?
Highly esteemed opponent, thank you for your nice words and for being here indeed after the Donders and now here in this setting. Thank you for your good critical question. So how strong is how convinced are we of this this one? I think for sure, or indeed we also talk in the discussion about this difference between the beta-1 and beta-2 receptors, and that we only inhibited one of them. So I think saying that the adrenergic system as a whole is not influencing the subjective emotion, I think that's might be a bit over ambitious. But I do think our results are, especially because we see it also in both drugs, so really, and they do show the results of, for example, the heart rate and the blood pressure, they do show that the drugs were effective, I think, at least in blocking this adrenergic system, because we see the output of an inhibited adrenergic system, and still the emotion or the subjective ratings are really the same and not a little bit different. They are really the same, I would say, if you look at the data. So that part, yeah, I'm convinced.
Yeah, okay. Yeah, thank you. So I think if you look in literature, many people would agree probably that just peripheral blockade would not have such an effect. The central one might be more contentious. And so I wonder, what is your reading of there's a lot of literature, of course, on effects of all kinds of adrenergic, noradrenergic agents on subjective experience of emotion, or perhaps an anxiolytic effect. So what is your opinion on the literature? Should we, because it has quite consequences, right? So if we say that these agents do not actually alter subjective experience, then we maybe should not be using them anymore for purposes like stage fright, or many examples where people use beta blockers, right?
Yeah. Yeah, I think, again, the same applies a little bit, this difference between the beta 1 and beta 2 and the beta system as a whole. I think the majority of the studies that do find results are inhibiting both receptors. So I think that would be very interesting in a follow-up study to test if there is a difference between, for example, propranolol that inhibits both, and, for example, metoprolol that only inhibits the beta 1 receptors. I think we need this kind of study to find out.
So you do think that the evidence for like beta 1 and beta 2 blockers, like propranolol, is sufficiently strong to make that claim, that it's clear that this does alter a subjective experience of emotion?
I think the literature is still mixed there. But I guess there's a reason that they do use it for stage anxiety.
I agree with you that if you look into the literature, that it seems kind of weak, right? The whole evidence for this, surprisingly weak.
Yeah, mixed. Yeah,
mixed.
But
okay,
in that sense, weak. Yeah.
And checking the time. So, could it also be that there would be a subjective effect, but you were not able to capture it? So, could there be something in the way we ask or we try to assess subjective emotion that the effect might not be accessible to the participant in the experiments that you do?
That's a very interesting question. I think that's, with null findings, I think that's always something we need to think about. Is it our experiment not picking it up, or is there no effect? If I would expect that to be the case for this panic induction, no, I do think also if we look at the meta-analysis that from what we have seen in the past that this panic induction it shows sensitivity to inhibition. So people do experience differences in the panic induction based on pharmacological effects, for example. So then I would say if there is an effect, why don't we? People would report it, I think, or we would see it in the data.
Yeah, okay. So you base on the findings in chapter 2 that there are many studies actually that do show effects directly on subjective experience in drug manipulations. Okay. I'm looking at the prorector if I...
Time is over.
Time's over.
Okay.
Hand the floor back to the prorector. Thank you very much.
Thank you for looking at me, but indeed the time is over. Thank you very much. So we proceed with Professor Jardy online. He is professor in child adolescent psychiatry at the University of Lille in France, and I would like to welcome him and give him the word to proceed. Professor Jardy.
Thank you very much, Mr. Prorector. As my colleagues, I wanted to start by thanking you for a very clear presentation and a very clear discussion of your PhD work. And I also wanted to congratulate you, notably for bringing to fruition an fMRI-based neurofeedback project. And we know the amount of work that represents, so congratulations again. And my question will mainly focus on chapter 6, as you can imagine, since I mentioned neurofeedback. And my first question relates to the effects of the graded neurofeedback procedure you described. And I wanted to know if, for you, what could be the potential reasons for the lack of differentiation between the medium target and the high target levels of insular modulation in this study?
Yeah, highly esteemed opponent. Thank you for your compliments, and great to hear that even for a non-Dutch speaker the presentation was clear. That's good to know. And thank you for your interesting question. So indeed, we decided to do this graded neurofeedback as we expect, this needs more control, or there's more specificity if we do see differences between the two levels. So why don't we see the difference? I think it might be just that it's too hard at this point, so that people are effective or they do succeed in activating or tapping into the insula, and on the group level they activated significantly, but that they didn't reach enough expertise or enough skills yet, or not yet, to differentiate between the two levels or to specifically activate it to the two targets. So I think for now more research is needed to see if people can do it at some point, maybe with more training, or for example continuous neurofeedback design, because now we chose to do it intermittently, but I think that can add some level of complexity for the participant. So I would be interested to see with the continuous neurofeedback, for example, or more training sessions, if people do achieve it at some point, or if it's always hard when to decide they are not able to. I think for now, I'm afraid it was too difficult or to reach this created influence on this study level.
Yeah, of course. No, absolutely. I really like your assumptions and last proposition regarding the maybe try to improve learning and also add some sessions. I think it's very important to be able to specify that. My second question was linked to the introspection measures. So I found this part also very interesting. And I wanted to ask you two questions about that. The first one is, could we expect such a difference between participants' estimation and the actual level of insular activity? So notably, if we think that you used a region of interest focused on the insula, do you think that other part of the network, for instance, could have been more interesting for that purpose, like the anterior cingulate cortex, which is known involved in monitoring, or other regions? Do you have any hints or any intuitions regarding that?
Yeah, sorry, I missed a little bit. So why we chose specifically the insula for interoception was your question?
Yeah, because you tested the relationship between the estimation of the participants regarding their performance and the activity level of the insula, which is a region of interest. But I was wondering, because you selected insula, but insula is also, if we look at the functional maps you present in chapter 6, we think that this is not the only area which could have been selected in the network. And I was wondering if regarding this introspection measure, other part of the networks could have been considered.
So sorry, I didn't quite, but I think indeed why the insula, or just, why did we choose the insula, especially with those research questions. So I think two reasons why we chose the insula. So the first one, really because of, also in line with the thesis, this region, I think literature shows, and a lot of people seem to agree, that it's one of the most important parts or regions for this interoceptive processing. And also indeed other regions are involved, of course, in this network. But I do get the idea from the literature that it's all connected to the insula at least, and also projecting via the insula. So that different regions or different activity or different information from different areas seems to converge to the insula or be collected in the insula. So I think, for interoceptive questions, I think it's a very interesting region and seems to be one of the key regions, I think.
Yes, I totally agree with that. I'm of course, I know that insula is a very key node in what you're interested in. But my question was more about the control monitoring that you expect the subject, so and the objective control they could try to do. So regarding the participants estimation, do you think that other part of the networks could have been also interesting to look at in terms of correlations between the subjective participant estimation and the control they are able to achieve in the neurofeedback procedure? But I totally agree with you regarding the major role of insula, and there is no doubt that the region of interest for the neurofeedback training has been well selected. That was not my point, but maybe regarding this very specific question, do you think that other part of the network could have been also considered?
Can you provide a short answer, please?
Yeah. Thank you for clarifying. I think that's always the tricky part with neurofeedback. Like you said, it is always a network. It would be definitely interesting to also dive more into what other parts of the network were involved and think about what should be targets in the future. Yeah, thank you.
Yeah, sure. No, that was a difficult question. Another point, if I may, was about the what this kind of result could mean in terms of blinding, because we know that there is also a huge issue in the neurofeedback literature about the need of more randomized control trials. So, and there is, of course, the need to use blind design. So, if people could have
I think we are
direct access.
Requested to continue with the next. Very sorry to interrupt.
Yes.
I'm sorry, Professor. I'm sorry, Professor Jardy. Wait, you switch off, maybe. I cannot reach you directly by our microphone system, so that only is possible here by the on-siters. We have to proceed, and we have still 3 more opponents to go, and I would like to go swiftly to Professor Schumann. She was also a member of your assessment committee, and she is professor in clinical applications of neuromodulation, and I'd like to give her the word.
Thank you, Pro-Rector. Well, yes, let me begin with also congratulating you. I think it's a fantastic and well-written thesis. I was very impressed. I was also impressed to see that parts of this thesis are already published. And of course, I'd like to extend my congratulations to the supervisory team. I think, like other opponents have said before, I think this combination of pharmacological, behavioral, and neuroimaging approaches is quite impressive. And I think it's really important for the field of effective neuroscience. So it's great to see what you did. Of course, I also have a question. And I'd like to, well, I have a more general question, but I'd like to link it to Chapter 5. Because in Chapter 5, you actually explored whether neural or behavioral indices of extinction learning could predict long-term outcomes of exposure therapy. But you didn't find a clear predictive value. We know that the ventromedial prefrontal cortex actually does play a role in extinction learning. And I'm wondering whether you think that non-invasive brain stimulation, such as TMS, could play a role here, that we could maybe enhance extinction learning and possibly improve therapeutic outcomes. If you think so, I'd like to know how you would design such an intervention and at what stage of the learning you would actually use this. Or maybe you disagree with me and think VMPFC is not a great site at all, but rather suggest another site within the salience network or default mode network.
Yeah, highly esteemed opponent, thank you for your compliments and your nice words. And a very interesting question, how to proceed from here. And can we, with non-invasive brain stimulation of the VMPFC, for example, enhance extinction learning? We're of course very interested in that question. I think it definitely has potential there. But like you say, it's tricky when should we apply such an intervention? Because indeed, we see, I think there are several studies that show, the more VMPFC is involved during extinction learning, the better the outcome. So based on that reasoning, we also expect, if there's more VMPFC involvement during exposure, we can expect the same results. We are testing. We see, one logical step to start with would be to do it before extinction. I think, like you've also written, another interesting thought maybe for TMS or non-invasive brain stimulation, it says also very important, what is the state of the brain at the moment that we do it? So another idea would be to do it first, do some level of extinction to get the area involved, and then maybe try to enhance it with non-invasive brain stimulation, for example, to see if that's effective. And other areas, I think, that would still be like the most logical option. Tapping back in the master thesis, I think there we looked at counter conditioning, or they looked at counter conditioning, and they saw involvement of more reward-related areas, for example, the nucleus accumbens or prediction error areas, to really counteract this fear memory. I don't think you can reach that with non-invasive brain stimulation, but theoretically it might be an interesting target as well.
Okay, that's very, very good, clear answer. So if because you also looked at networks, right? And if I were to stimulate, for example, VMPFC with an excitatory protocol, I actually enhance activity ideally of the VMPFC, but I might have an enhancing effect on the entire network. Yeah. I think you looked at some data and you actually found anti-correlations. So could this be a problem, that if I enhance VMPFC that I actually have an enhancing effect on the entire network, or don't you think this is a problem?
Let me think about that. So the problem, if it acts on other parts of the network in a way that we don't expect it to, then maybe in that line of thinking, again, it would be worthwhile to think about what is the VMPFC doing when we try to stimulate it. I think the general idea is for extinction learning that it's inhibiting the amygdala. So if we can strengthen this inhibition, which I think would be achieved by, or could be achieved by stimulating the VMPFC itself, then I think that is a desirable connection that we want to influence. But if there are other connections that are also influenced, which we don't want to influence, that's something to carefully look into. What areas are exactly connected to the VMPFC and how that might interconnect. Yeah.
Thank you. Do you have time for another one or not? Do you think if we combined TMS and fMRI simultaneously, could this answer the question that I just asked? Or would we learn a little more about that then?
Yeah, definitely. I think there are already a lot of data on those extinction paradigms. So maybe if we specifically look at those data with that question in mind, we might find a better answer. And of course, doing it simultaneously will give us more insight in what exactly it is target, or if we do it during extinction or before extinction, how exactly is it targeting the network? For sure. I think that's will provide us with more information.
Thank you.
Sure. Okay. I have a bit of a longer one, but a short one. In chapter 4, you actually didn't observe task-related changes in the salience network, which was rather surprising, right? So I was wondering why? Why do you think you didn't find anything, and what would you change in a future study?
So
a long one. Sorry.
Thank you for the very interesting question. Yeah, that's also something we kind of hypothesized to find the salience network involved. So our discussions came out to, maybe because the fear conditioning task, it's also quite to some extent a cognitive task. You need memory, you need attention. So maybe there are just too much other processes going on for the salience network, or overshadowing the salience network. Or like we say, maybe the stimulus itself, a shock, it's unpleasant, but people know it's not harmful, it's a safe environment. They know they go out of the scanner and go home. So maybe really the nature of the task, or also the timing of the task, it's quite fast after each other. Maybe that explains why we didn't see salience network involvement.
Okay, thank you very much. I'll give the word back to the prorector.
Thank you very much. Now the opposition will be continued by Dr. Kwaadvlieg. She's affiliated with the Department of Neuropharmacology and Psychopharmacology at our university, and I'd like to give her the word.
Thank you, Pro-Rector. Dear candidates, I also would like to compliment you with this well-written thesis. And as said before, it really includes a lot of methods. And I think that reflects also the background of your promotion team, but also really shows that you have now a lot of scientific skills and competences. So congratulations also to your promotion team. I want to start also with diving in chapter 3 a bit more. And then first more focusing on the design. So on page 65, you wrote that the degree to which people experience panic symptoms, subjective panic symptoms, will differ between participants. And this is also the reason why you did a pre-screening session, and then based on that session, you selected participants on a criterion. I couldn't find the data of this pre-screening session in your thesis, but I was wondering, could you recall a bit like what kind of subjective values were participants experiencing during this first session of the CO2 inhalation?
Esteemed opponent, thank you very much for the compliments and this very interesting question. And is your question about the people that were selected, or just everyone?
That were selected. Yeah.
That were selected.
So what kind of, if the scale is from 0 to 100, right?
Yeah.
Do they score the first time at the 10, 20, 40, big kind of a feeling, just doesn't have to be exact numbers?
The exact numbers, indeed, it's a while ago, but I do remember, and I think that we also see this in previous studies, that repeating the inhalation by itself might diminish a little bit, but it's quite consistent in within a participant how the experience is. So I think, at least for the ratings, this should, I think it's comparable, but also with the data that is in the thesis, so the data of session 2 to 4. And
then you nicely immediately already tap into where I actually do want to go. So repeating the inhalation in the same participant multiple times. I was a bit wondered about your statistical analysis. So you say like it's based on previous studies. I also looked a little bit into the literature. Most of the studies are quite old, I have to say. But I do think that there are quite some studies actually showing that there is sensitization or habituation in a subpopulation of participants. So I had expected actually that in your statistical models that you would include order of conditions to just see like, okay, if they would have gotten a certain pharmacological manipulation in the first session, does that matter versus the fourth time? Is it correct that you, did you look at it? Maybe I missed it.
No, not at the thesis. So, the order of pharmacological manipulations was counterbalanced, so at least, the effect, or like I think the interaction between a certain manipulation and the session, I don't expect that shouldn't be there because of the counterbalancing, and just an order effect in general. We didn't report. I think we did look into it. But as I said, I think that's quite consistent. It's not that per se like the first session. There's differences disregarding the pharmacological manipulation, I don't think there are differences between the sessions.
No,
If
okay,
that's your question.
Because I think what has also already been discussed before, there are null findings regarding the subjective effects, and of course that's a bit disappointing.
Yeah.
So if I come back to this first point also, like whom you included, so the criterion that was used was like a 25% increase on this VAS scale, so for fear and discomfort. So if I would take two participants, and one is having a baseline 10 score and the other one 50, they both increase 25%, so then you 12.5 and 75. I'm wondering how you, the increase is the same, so you would have included them. I think that is also what we see in the violin plots. So the violin plots show quite some variation in the range. So this is on the, if you know the page better, I guess, than I do. I have it written down. But anyway, the subjective change, I think, is quite different for someone going from 10 to 12.5 on a fear level. In my opinion, they still don't really experience fear, while someone who moves from 50 to 75, they were already at baseline experiencing quite some fear, and are now for sure really after the inhalation experiencing fear. Do you share that this might be an issue, also thinking about the null findings, how you defined and included your participants? And if so, would you now change maybe the criterion?
Yeah, very important question, of course, how to optimize or how to design the study. Yeah, I do agree there might be a subjective or experience difference if someone scores at the end 75 compared to 50, either if the relative increase is the same. I think because of the analysis we do, it's like within subject, so if there would be differences between the conditions, I think we still pick it up because of this within subject design. So for the relative differences within a person between the sessions would then
But what do you think of the meaning? Because if it's someone that it's like around 10, do you then think like that we're really talking about fear, experience of fear symptoms?
No, so yeah, at least they need to score 25 post, like if they would have started from 0. Yeah, is that meaningful, or is it as meaningful as someone from 50 to 75? I think that's a very good question. I think, yeah, it's for sure a good start, because if we didn't do this screening, then we also included people that didn't increase at all, because some people said, oh, this was fun. And yeah, I think very important question. I don't know the direct answer, I think.
Let's discuss it maybe for follow-up.
Yeah, no,
I will give the word back to the prorector.
Thank you very much. Then we proceed to the last opponent. And again, he will get full time too, so don't worry about this. Dr. Hernaus, he is affiliated with the Department of Psychiatry and Neuropsychology, and I'd like to give him the word.
Great, thank you, Prorector. Dear candidate, I guess at this point there are very few things I can still say that haven't been said by the others. I fully share with them that I read your thesis with great interest, great enjoyment. Like the others, I agree that I think what is a major strength of this thesis is this nice coming together of all these different techniques in the service of like true experimental work, which is a passion of mine. So I fully appreciate this, but maybe I can say something that the others haven't said. And what I appreciate in the thesis is also this ability of yours to place your own work kind of within a historical perspective. I think I see this in the propositions, in the introduction, in some of the chapters, and I can appreciate it when someone has this historical awareness of the work that has become before them. So major compliments to you and to your esteemed team of promoters. And that brings me to my questions. And just like the others, I would also like to start with asking a question about chapter 3. And I think this is a bit of a fundamental interpretation question, because I was looking at your results, and I turned to page 71, which is the final page of the results. And there, I think almost in a byline, you mentioned there at the bottom that induced changes in physiological and subjective parameters do not seem to be correlated. And here I'm thinking you have done this study to manipulate particular physiological or peripheral mechanisms, assuming that they contribute to the central experience of emotions. But I think what you mention here is that there's actually no relationship between the periphery and the central processes, which makes me wonder, does this pull the rug from underneath the chapter here? Does this change, affect the central logic behind the thesis? The chapter, I mean, sorry. Your answer?
Esteemed opponent, thank you for your kind words and for the appreciation of the historical perception. And thank you for your question. I'm not sure if I fully understand. So yeah, that we don't see the correlation between the parameters. How that influences. I think, if any, it's in line with the conclusions that we draw. Or but one thing we're testing if this reduction in physiological parameters is related to a reduction in subjective parameters. So it's at least in line with that, I would say.
But I think you're building this thesis on the assumption that the periphery contributes to the subjective experience of emotions, no?
That's one of the questions we try to find more. I think this is, indeed a very common assumption, and I think I would intuitively say that's true, but I think the data are quite mixed on that, and that's also what we try to contribute to.
But I think there might be an important distinction, because I think you might be talking about the effect that the pharmacological manipulations don't affect the endpoints, but I'm saying the peripheral and the central endpoints aren't actually, they're not even related. So the heart rate, the blood pressure you've collected, they're not even related to the ratings people give. Mm-hmm. And I think that's a fundamental, is that not a fundamental problem? Because I think you in your introduction, you write that these things contribute to the experience of emotions.
There's a lot of theories that state that, and that's one of the questions we try to disentangle further. And at least based on the results in chapter 3, we don't find direct, at least, support for this relation or this contribution of the physiological parameters to the subjective parameters.
So what do you think here? Do you think you've disproven the theory that peripheral sensations contribute to central experience and emotions? Is that the evidence in the?
No, I would not state that. What I write in the discussion, I think there's that indeed is too simple to say, especially also with the whole history. We cannot just and we see this a little bit in chapter 3, but I think there's more to it. So other questions or alternative explanations for just not having this relation could be, we measured heart rate and blood pressure. They were lower, but for example, if you looked at the relative increase of heart rate, we didn't see differences between the conditions. So maybe this relative change in physiological parameters is more important than just, also is similar to what's the meaning of values on their own. You always need to consider them relatively to someone's baseline or someone's starting point. So I think that's something that really needs more looking, and also more reporting in studies that are being done. Because I wouldn't disregard the influence of physiological.
My reading. Do I still have time for a short question? My reading of the chapter was perhaps that maybe the peripheral phenotypes were were ill chosen because they don't relate to the central experience. I would just, as a final question, maybe looking forward, do you think there are better peripheral markers out there of emotions? And if so, what could they be?
Yeah, I think definitely for panic, I would be very interested to look into the respiratory parameters. Also, it's maybe difficult because it's intertwined with the CO2. It is a respiratory challenge. But I think, that would be very interesting for panic at least to tap into. And I think previous results have also shown that, for example, heart rate variability is more, or tells different information. So those two I would like to look into.
Perfect. I'm slowly going to give the word back to the pro-rector while we wait for the final words.
Basically, there is no need to give the word back because the pedal is really closing here the session. So I switch back to Dutch now, okay? Geachte kandidaten, geachte mevrouw de Vos, de gelegenheid voor het verdedigen van uw proefschrift is thans voorbij. De promotiecommissie zal zich nu voor beraad terugtrekken en ik verzoek u en uw gezelschap onze terugkomst en de uitslag van ons beraad in deze zaal af te wachten. Wilt u gaan staan?
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Southside, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Southside, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Geachte mevrouw de Vos, de commissie hier aanwezig heeft zich uitvoerig beraden, niet zozeer over de kwaliteit van die proefschrift, want dat was reeds goedgekeurd, maar wel over de wijze waarop u dit proefschrift hedenmiddag heeft getracht te verdedigen. Op grond van een positief oordeel en gelet op de examens die u vroeger heeft afgelegd, heeft ze besloten u het gevraagde doctoraat toe te kennen. De hoogleraar professor Linden is gemachtigd u op de gebruikelijke wijze met deze waardigheid te bekleden en ik geef daarom het woord aan uw promotor.
Belooft u dat u altijd volgens de beginselen van wetenschappelijke integriteit te werk zult gaan, eerlijk en zorgvuldig, transparant, onafhankelijk en verantwoordelijk?
Dat beloof ik.
Krachtens de bevoegdheid ons door de wet toegekend, volgens het besluit van de commissie hier tegenwoordig, verklaar ik hierbij u, Jette Hanna de Vos, tot dokter te bevorderen en u alle rechten te verlenen die daaraan volgens wet en gewoonte zijn verbonden. Ten bewijze hiervan overhandig ik u nu de bul door rector, secretaris en overige leden van de promotiecommissie ondertekend en met het grootzegel van de universiteit bevestigd.
De kandidaat, of de jonge dokter moet ik beter gezegd zeggen, nou, de jonge dokter krijgt nog meerdere andere certificaten en het volgende certificaat wordt overhandigd door professor Schuurs. Zonder bril. Okay.
Dear Doctor, on behalf of EURON, that's the European Graduate School of Neuroscience, I am entitled to hand out the official EURON certificate. EURON is a training network of 7 universities in the Netherlands, Belgium, Germany, France and Luxembourg. And this certificate is awarded to PhD graduates that have participated in multiple courses and conferences of the EURON program and have collaborated and worked in a foreign research group and have published their work in international scientific journals.
En vervolgens is 3 maal scheepsrecht, dus dokter Leibold gaat nu overhandigen het zogenaamde menscertificaat.
On behalf of the Research Institute of MENS, I'm entitled to hand out this official certificate. The owner of this certificate has received training in a broad range of academic skills and has gained experience that is relevant for the successful career as an independent scholar.
De laudatie vervolgens zal worden uitgesproken door dokter Leibold.
Beste Dr. de Vos, beste Jette. Ik heb de eer om een van de eerste te zijn die je mag feliciteren. Proficiat met het behalen van je doctoraat. En felicitaties ook aan je familie en je partner René, die je gedurende deze reis hebben gesteund en begeleid. 5 jaar geleden begon je reis hier en een nieuw hoofdstuk. Het begon met een interview voor een promotieplek. Wij waren al heel snel van jou overtuigd. We zagen een perfecte match. Het wordt duidelijk dat je bij het Donders Instituut al veel ervaring had opgedaan in het kader van emoties en dat je het potentieel had om te groeien en te leren. Jezelf twijfelde wel nog een beetje of je dit avontuur echt wou beginnen. Maar gelukkig ben je uiteindelijk wel naar Maastricht verhuisd. Naar een stad die je nauwelijks kende en dat midden in de coronapandemie. Corona maakte jouw start bij ons niet gemakkelijk. Maandenlang had je nauwelijks kans om je collega's te ontmoeten en te leren kennen. Vergaderingen waren online en als we elkaar op Vijverdaal ontmoetten, dan zaten wij met z'n tweeën met mondmaskers op in de kelder in het CO2-lab, zonder iemand anders tegen te komen. Dus hoewel de omstandigheden ver van ideaal waren om nieuwe technieken en vaardigheden te leren, heb jij dat gedaan alsof er gewoon niks is. Je leerde heel snel hoe je de CO2-inhalaties en de fysiologische metingen kon doen. Binnen enkele weken had je een volledig ethisch protocol geschreven en in een kort tijd je eerste studie afgerond. Je hebt een groot aantal proefpersonen kunnen recruteren en herhaaldelijk kunnen testen, zonder problemen. Er zijn maar weinig mensen die dat op die manier kunnen. Zeker niet aan het begin van een promotietraject. Bij de eerste studie zou je nog kunnen denken, een beetje geluk gehad, alles is goed gegaan. Maar toen kwam de tweede studie, de volgende analyse, de derde studie, nog een analyse. Of het nu ging om onderzoek met gezonde vrijwilligers of met patiënten, ingewikkelde neuroimaging-analyses of het leren en toepassen van neuromodulatie. Alles verliep soepel. En toen werd nog duidelijker dat een beetje geluk gehad een naam had. Jette. Jouw aanpak leidde tot succes. En vandaag staan we hier met dit prachtige proefschrift dat jouw successen toont. Een verzameling aan studies over body, brain en emotions, zoals je ons vandaag hebt laten zien. Maar deze titel gaat verder dan het onderzoek. Het is ook een overkoepelende beschrijving van jou als persoon. En van alles wat je de afgelopen jaren hebt meegemaakt. En ik zal jou een aantal voorbeelden geven. Laat mij beginnen met het brain, de hersenen. Zoals ik al zei, je leerde alle nieuwe technieken supersnel. Je hebt de afgelopen jaren laten zien hoe intelligent je bent, hoe zelfstandig je kunt werken en hoe betrouwbaar je bent. Ik hoefde je nooit iets 2 keer te zeggen. Je werkte gestructureerd en hield overzicht. Een eigenschap die vandaag speciale aandacht verdient, is je bescheidenheid. Je bent de meest bescheiden persoon die ik ooit heb ontmoet. Je staat altijd klaar voor je collega's, je lost moeilijke problemen op en je laat anderen nooit het gevoel krijgen dat je het beter weet. En daarom liepen veel collega's ook bij je binnen om advies te vragen. Je bent voor velen inmiddels een rolmodel. Ik wil worden zoals Jette, heeft letterlijk iemand tegen mij gezegd. Jouw bescheidenheid kwam ook vaak terug in onze labmeetings. Op het einde wordt altijd gevraagd of iemand nog iets wil delen. En jouw antwoord was bijna altijd, nee. Maar meer dan 1 keer begon ik opvallend te hoesten. Ja, om erop te wijzen dat er toch iets was wat ik waardevol vond om te vermelden. En jij wist altijd meteen, zonder na te denken, wat ik bedoelde. Bijvoorbeeld dat je net een manuscript had ingediend, dat je proefschrift naar de leescommissie werd gestuurd of dat je goedkeuring had ontvangen. Ja, en dit zijn geen kleine successen, maar jij vond het niet nodig om dit in de groep te gooien. Ja, en daarom kreeg je ook de titel Queen of Underselling. Of zoals we soms zeiden, hier is understatement en hier is Jette. Natuurlijk kreeg je na die subtiel afgedwongen updates best veel aandacht van ons. En er stond zelfs het vermoeden dat je soms expres iets niet vertelde, omdat je het stiekem toch wel leuk vond om achteraf extra veel aandacht van ons te krijgen. Ja, komt het bekend voor thuis? Je bescheidenheid kwam ook terug in je sportprestaties. Je hebt de afgelopen jaren je body, je lichaam, goed laten werken. Je hebt spieren leren kennen waarvan je niet wist dat ze überhaupt bestonden. En het begon met René. René houdt van sport en jij begon mee te gaan lopen. Een kort stukje werd een langer stukje, fietsen en zwemmen kwamen erbij. En uiteindelijk deed je ook mee aan wedstrijden, met korte en langere afstanden. Ja, kijk, in een zwembad te zwemmen, dat heb ik al vaker gezegd, is 1 ding. Maar jij zwom ook in het open water. Koud water, je ziet de grond niet, andere gemotiveerde deelnemers die ellebogen uitdelen en ook nog golven maken. Wat een prestatie om aan zoiets deel te nemen en dan ook nog de finish te halen met een goede tijd. En wat zei jij daarover? Heel bescheiden. Oh, het doel was het om het te halen. En je liet achterwege hoe goed je het had gedaan. In de afgelopen jaren heb je ook veel met emoties te maken gehad. Zowel in het onderzoek als privé. De cover van je proefschrift straalt rust uit. En dat weerspiegelt ook jouw persoonlijkheid. In elke situatie bleef jij rustig. Tijdens het testen van deelnemers, bij onverwachte resultaten, tijdens presentaties op symposia. En zelfs tijdens een vakantie toen jullie in een tent moesten slapen en het heel erg veel regende. Jij bleef rustig. Het duurde lang voordat ik je eens een beetje zenuwachtig zag. Namelijk tot de dag dat jouw proefschrift naar de leescommissie werd gestuurd. En zelfs toen was minstens 1 van je 2 paranimfen zenuwachtiger dan jij. En gisteren ook en vandaag ook, denk ik. Tijdens je promotietraject heb je ook René leren kennen. Jullie hebben jullie gezin uitgebreid met 2 katten en een huis gekocht. Begin dit jaar hoorden we dat jullie gingen trouwen. Maar natuurlijk vertelde je dat niet direct aan de hele groep. Ik zal die situatie even samenvatten voor je familie. Dus we zaten aan een aantal tafels en er vonden meerdere gesprekken tegelijkertijd plaats. En jij zat ergens zo aan het uiteinde en plots ging er een golf over de hele tafel heen. Jette gaat trouwen, Jette gaat trouwen. Wat? Jette gaat trouwen. Jette, vertel! En meteen was er alle aandacht op jou. Je werd duidelijk gelukkig van al die mijlpalen in je privéleven. En ik hoop dat je ook blij werd van het samenwerken met ons, want wij hebben enorm genoten van de tijd met jou. Van jouw team spirit, dat je ons spelletjes als Exploding Kittens leerde, en dat als kattenliefhebber. Je droge grappen die vaak heel onverwacht kwamen. En jouw niet-verbale manier van communiceren, waarbij zonder woorden heel snel, heel duidelijk is dat je er niet mee eens bent. En we hebben genoten van jou als persoon en onderzoeker. Jette, je hebt het echt wel verdiend om hier vandaag te staan, om je doktertitel en de mens- en Euron-certificaten te behalen. We zijn blij dat we jou mochten begeleiden en dat je nog een aantal jaren bij ons blijft werken. Je gaat nu met de politie aan de slag om agenten weerbaarder te maken. Wat een mooie studie om je werk voort te zetten. Ik wens je heel veel succes en alles wat nog gaat komen op het werk en privé. Proficiat. Ik het woord terug.
Beste jonge dokter, hoe voelt dat? Zo te zien aan de body language voelt goed. Ja?
Zeker.
Vol emotie. Ja, om het maar eens aan te reflecteren aan het proefschrift. Ik wil je van harte feliciteren, ook namens de Universiteit Maastricht, namens onze Board of Directors. Je hebt het prima gedaan en ik kan zeggen dat wij in ons beraad tot een unaniem positief besluit zijn gekomen. Daar was geen twijfel over mogelijk. Ik wil in mijn gelukwensen ook betrekken het promotieteam die jou de afgelopen 5 jaar begeleid hebben. En ik denk, dat is eigenlijk nog niet echt zo naar voren gekomen, vind ik zelf, is. Je hebt natuurlijk gepromoveerd in de coronaperiode. Dus je hebt echt een heel stuk werk thuis moeten doen of vanaf een computer moeten doen. Eenzaam, nou niet helemaal, ik denk dat René in de buurt was, ja, om dit proces te begeleiden. Maar in ieder geval, ik wil het promotieteam die je ook in deze moeilijke periode hebben begeleid, ook van harte danken namens de Universiteit Maastricht. In mijn gelukwensen gaat natuurlijk de familie die hier voltallig in de eerste rij aanwezig is. Ik denk gedeeltelijk uit Dordrecht, heb ik zo gelezen. Dus ik denk een hele rit vanuit Dordrecht. Ik wilde ook bedanken de leden van de corona die toch weer de moeite hebben genomen om te reizen, te lezen, vragen te bedenken, hier naartoe te komen enzovoort. Hartelijk dank, ook namens de Universiteit Maastricht. En met deze woorden wil ik dan dit sluiten. En voor het afronden van deze officiële ceremonie gebruik ik mijn hamer, die jullie nog niet gezien hebben en die ik nu zal laten zien. En hierbij is deze zitting dan officieel gesloten. Hoe gaan we nu verder? Er is zo meteen een receptie in onze zogenaamde refter. Dat is de ruimte die, als je naar buiten gaat, aan de rechterkant is. Wij gaan hier nog een foto maken, samen met de 2 leden van de online commissie, die nu op het scherm aanwezig zijn. We maken ook een foto op de trappen van de universiteit. En daarna vervoegen wij ons ook tot de refter in ongeveer 7 à 8 minuten. Ja, dat is de timeplanning die van ons hier bestemd is. Goed, dan zou ik zeggen bedankt en tot zometeen.